Hey, good morning, everybody, and welcome. Very pleased to see so many of you here this morning. I would like to start off with the recognition, the land recognition of the territories on which York University resides. So York University recognizes that many Indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Tekaronto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, and the Huron-Wendat. It is now home to many First Nation, Inuit, and Métis communities. We acknowledge that the current treaty holders, the Mississauga of the Credit First Nation, this territory is subject to the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. So I want to spend just a few minutes telling you very briefly about the group that is bringing this event to you, the Gairdner Foundation, and many of you probably know little, if anything, about the foundation. It was founded in 1959 by a man by the name of James Arthur Gairdner, who was convinced by friends and family to put his considerable wealth to good use. And that good use, in his view, was the recognition of excellence in scientific endeavor that leads to good for society. So a main mission is to bring people like you here to hear about excellence in science and be excited and think about this as your futures. The awards, there are a number of awards. There is the Canada Gairdner International Awards, and our two speakers this morning are laureates, are recipients of this award this year in 2022. There is the Canada Gairdner Whiteman Award, recognizing Canadian medical scientists and administrators, and the John Dirks Canada Gairdner Global Health Award. These are Canada's most prestigious awards in biomedical science and rank amongst the most prestigious scientific awards in the world. They're unique in many ways, including what we call the national program that we are part of today. It brings present and past award winners to speak about their award-winning science at venues across the country. At many of those venues, we speak to groups like yourself, like young high school students, and the mission of this program is to put scientific excellence at the forefront of Canadian science. So Gairdner events are a true Trans-Canada celebration. This week, the laureates have been from Vancouver to, I think they're in Quebec City at Laval. Today, some of our laureates speaking. And there are also a series of exciting symposia. And last night was a phenomenal black tie gala awards dinner where the laureates received the recognition and their awards. So this presentation at York is part of the Gairdner National Program for many years, and we are delighted to have the opportunity of partnering for this exciting program. And York has been involved with the Gairdner program, I believe, for 18 years now. I'm Ron Perlman, emeritus professor in the Department of Biology, and I've been involved with the Gairdner Foundation and with the York program for actually more than those 18 years, but with the York program for its existence. And the idea here is not to teach science. This is not a science class, but to use award-winning science to talk more about the scientist, how they decided to go into science, who their mentors were, the excitement of doing science, and providing advice to students to excite the students about biomedical science. So the Gairdner, there is another series of talks this afternoon, which are research talks to faculty and trainees about details of the science that the laureates have done. As some of you may know, the Gairdner Awards are excellent predictors of Nobel Prizes. So nearly 30% of Gairdner Award winners have gone on to receive Nobel Prizes. And that number, I believe, is now 96 Gairdner laureates have gone on to receive Nobel Prizes. And many of those people have been speakers to this group at York. So it's an exciting recognition, and it says something about the Gairdner process of selection of laureates, in that we're exceptional predictors of future recognition by our laureates. So again, it's my pleasure to welcome you here today. Thank you for coming to this exciting program, which I'm sure you'll enjoy. These programs cannot take place without our sponsors, and it's not up here right now, but on the loop you have seen some of our sponsors: the Government of Canada through the Canadian Institutes of Health Research, the Globe and Mail, and York University. And the sponsors are instrumental, and we couldn't do these programs without them. So now I'll ask a student, honors biology student at York, Brittany Remnant, to introduce our speakers. And she'll start with the first speaker, then we'll have a Q&A after that, and then she will introduce our second speaker. So, Brittany.
Hi, everyone. I'd like to echo Dr. Perlman in welcoming you all to the proceedings of the 2022 Canada Gairdner Awards program at York University. My name is Brittany Remnant. I'm a fourth-year biomedical science student, and it's my honor to be introducing two of the pioneering speakers who have been selected this year as recipients of the 2022 Canada Gairdner International Awards. Now, before introducing our speakers, I'd like to take a moment to address our high school audience. And having been in your shoes only a few years ago, I know it may seem like participating in primary scientific research is out of reach, or like it may be an eternity away. And I want to tell you that that doesn't have to be your reality. There are many opportunities for undergraduate students to partake in research as early as your first year. You can volunteer in research labs, you can enroll in research practicum courses, you can apply for summer research awards. The opportunities are there for you, you just have to look for them. I decided to get involved in research because I've always known myself to be driven by an intellectual curiosity of the world. I want to develop the knowledge and skills to be able to investigate and answer my own questions. This past summer, I completed a Dean's Undergraduate Research Award, and I'm currently working on my honors thesis project in the Bayfield Lab, where I'm studying protein-RNA interactions and alternative pre-tRNA processing. I find being in a lab space to be invigorating. There's nothing quite like having a question, thinking through how you may be able to find an answer, probably failing again, and then maybe finding the answer you were looking for. Maybe you'll find something you weren't looking for at all, and maybe as was the case for our laureates today, you'll find something that will be able to help others along the way. I'm hopeful that your being here today reinforces the perspective that research is an attainable possibility, and I encourage you to actively pursue your interests as you move on in your post-secondary studies. Don't hesitate to both seek out and pave new opportunities for yourself. I hope you walk away today inspired by the amazing work of our laureates and excited by your own future and potential. So with that, I'd like to introduce our first speaker, Dr. Stuart Orkin. Dr. Orkin is an esteemed physician and stem cell biologist whose research is focused in hematology and more specifically the genetic basis of blood disorders. Dr. Orkin completed his undergraduate studies at the Massachusetts Institute of Technology, his medical doctorate at Harvard Medical School, and postdoctoral research at the National Institutes of Health. Dr. Orkin is the David G. Nathan Distinguished Professor of Pediatrics at Harvard and is an investigator at the Howard Hughes Medical Institute at Boston Children's Hospital. Dr. Orkin has received several distinguished awards over the years, some of which include the William Dameshek Prize, the George M. Kober Medal, and the Harrington Prize for Innovation in Medicine. Dr. Orkin is a recipient of one of the Canada Gairdner International Awards this year for his groundbreaking discovery of the protein BCL11A's role in suppressing fetal hemoglobin genes in adults. Dr. Orkin found that suppressing its gene can dampen the symptomatic severity of hemoglobin disorders. He's currently conducting clinical trials to test this novel treatment method. His preliminary gene therapy trials have been able to prevent sickle crises and anemia in sickle cell patients and have limited the need for blood transfusions in patients affected by beta thalassemia. These hemoglobin disorders affect more than 5 million people worldwide, and as such, this novel gene therapy treatment could have profound impacts for affected individuals and will likely contribute to making the treatment more accessible for a greater distribution of people. So please join me in welcoming Dr. Stuart Orkin.
Thanks very much for the kind introduction, and I'm very glad to be here and see, young scientists or budding scientists. And what I'll do first is tell you a little about how I sort of got to where I am and what I do, and then tell you some about the work we've actually done. So I like this quote from Goethe, which is, blood is a very special juice. And I've had an obsession with blood for, 40 years. And maybe, around time of Halloween, it's a good time to think about blood. And, So who am I and how did I get to where I am? Well, I'm what's called a physician scientist. So I'm someone who's trained in both medicine and science. So that means it's a long path of training, but one that brings really biology and science directly to bear on patients and their diseases. I specialized in hematology and oncology. That's the study of blood and cancer, not just blood cancer, but also cancers of solid tissues. And I work in an academic medical center, which is an institution that's broadly committed to excellent clinical care, advancing that care, developing new science and discoveries that could be applied to patients. And these are very unique institutions that are different, at least in our country, the United States, are different than community hospitals, which basically deliver care and don't specialize in finding new knowledge or teaching sort of the next generation of physicians. So it's a mission that's it's clinical care, research, teaching, all goes on in the institutions. And the training path that I've had includes medical school with or without a PhD degree, and postgraduate, both clinical training and scientific training. I'm an MD. I do not have a PhD, and in part, that's an accident of the time I trained, because at the institutions I trained, and particularly the Harvard Medical School, they actually prohibited getting 2 degrees. That's very unusual now. Most institutions are glad to give you 2 degrees if you'll go through the training, but at the time they said, well, 1 degree was enough. You didn't need 2 degrees. It's a little different now. I think many physician scientists will have both degrees. So how did I actually get into biology? When I was a student at MIT in Boston, I thought I was going to be a physicist. I was very good in math and physics, but I didn't know anything about biology. Biology really was like dissecting frogs from my high school experiences. And When I was at MIT, I realized that there were lots of other students who were very good in physics, so I wasn't that unique. And I took introductory biology that was taught by this gentleman here, Salvador Luria, who received a Nobel Prize, but after I took his course. But he was one of the founders of really molecular genetics. He's best known for really showing that mutations are really random genetic events. And he was an inspiring teacher. And fortunately, at MIT, the most senior faculty would teach the undergraduates, which is not always the case in universities. After my medical school training, I went to the National Institutes of Health in Washington, Bethesda, Maryland, where there was, at that time, a program for training physicians in science. And this was part of the Public Health Service. And that sort of period was a sort of the high point of training at the NIH for physicians. And partly it was because at that time the academic medical centers were not as developed as they are now, so this was really the center of activity. And there I trained with Philip Leader, who was one of the founders really of molecular biology and gene cloning, best known for one of the the early pioneers in cloning of genes, and also for demonstrating that genes that are involved in antibody production, the immunoglobulin genes, are rearranged during the process of development of lymphocytes. And at the NIH, what I learned from laboratory and from Philip Leader was to focus on a problem and not get too distracted sometimes by the kind of things that happen that aren't necessarily central to your problem. And then I returned to Boston for some additional training and came under the mentorship of another senior individual here, Dr. David Nathan, who's now 93 years old and still active. And he's one of the sort of pioneers of blood cells in the United States, and particularly in training generations of physician scientists. And he's really provided an incredible environment for training and for discovery. So I've had a long career, and I'll just tell you some of the phases that I've sort of lived through and experienced, because I think it'll make a point about how you have to think about a career. I've lived through many new areas of biology, and the first one was recombinant DNA, or gene cloning, which became first really possible in the early 1980s. And really, when I established my own research laboratory, I had to learn gene cloning and apply it to the problems I wanted to study. So I really had to learn new technology as it came on the scene. What that led to in the discoveries, one of which I'll sort of summarize in a little bit, was the ability to transfer some of that science into clinical practice. And that was through prenatal diagnosis using DNA. And this is the diagnosis of disease in a developing fetus before birth, which has been very useful in many circumstances. After that, I actually became involved in another area called positional cloning. This is actually cloning a disease gene without really knowing much about the product of that gene. And that's been a field that's really exploded in human genetics. Genes such as that for cystic fibrosis were cloned using that kind of approach. And that was in the mid-1980s. Other work that we did led us into stem cell biology or regenerative medicine, where one studies how cells give rise to more differentiated cells in the body. And that's a field that's exploded over the last 2 decades. And I also became involved in another field called genome-wide association, which is a field that's exploded in human genetics, which is a study not of rare disease, but of common genetic variation. And I'll just give you a sense of that as well. And then finally, over the last 5 years ago, we've become deeply immersed in the field of genome engineering, gene editing, CRISPR, which I'm sure all of you have heard about. So, sort of what take-home messages do I have for traits for success? And, you know, Katalin may have similar suggestions when she talks. The first one, I think, is do something you're interested in. If you're interested in it, you'll want to do it, and if you're not interested in it, you won't pursue it. So, and you'll enjoy what you do, which is really important, I think. One of the gifts of a career in science is you get to explore, use your curiosity, and you can have a career that is fulfilling, gratifying, and you have enjoyment, and you actually get paid to do it, which is really a gift. In many areas, you do a job, you get paid, but it's not a job you really want to do. So this is a real perk of what we do. Focus on some goals. You want to really know where you're headed and try to attain those goals. You have to manage your time, and this is something that I'm sure you all recognize. There are many different things you can do, many activities, both in and out of school. I think it's important to learn how to manage your time so you can begin to think about what goals you have. And prioritize, because there are too many things to do and you can't do everything all at once. We all think sometimes we're supermen or superwomen, but it's hard to do them all. Importantly, remain flexible, adaptable, because in a field of science, there are new discoveries, there are new technologies, new approaches, and you can't stay with the same one for an entire career. That's not particularly interesting. Sometimes if you do that, you can reinvent yourself as you go along. And learn from others and collaborate. Science is a collaborative activity, it's an interactive one, and you can't do it all yourself. And don't get discouraged. Failure is part of life. It's a learning experience, and we don't like to fail too many times, but you do take away lots of positive things from every failure. So I'll just tell you a little about sort of what we've done and try to give you a sense of how we've discovered things really through our own curiosity and also been able to apply some of this, I think, to the benefit of patients. So I've been interested the entire time in blood cells. I want to know where they come from. How do you develop a blood cell? How do they go awry in disease? And then if you can figure that out, can you do anything about that disease? Can you treat patients, develop new approaches to therapy? And so where do blood cells come from? Well, they all come from what are called hematopoietic stem cells or blood stem cells. These are the cells that actually allow you to perform bone marrow transplants, which are common clinical procedure now in a number of situations. And here you can take cells from an individual, give them to another individual, and regenerate the whole blood system. And these stem cells give rise to red cells, white cells, which are used in host defense, and platelets, which are important for basically stopping bleeding. And blood cells have a hierarchy, and this is a cartoon from the New Yorker magazine showing the sort of ice cream at the top and what you can do with ice cream in different ways. But this is sort of the science view of a stem cell, which ultimately gives rise to all these mature cells shown on the right. And these mature cells have all different functions, all of which are critical to sort of normal health. And the blood cells can go awry in disease in a number of ways. You can inherit mutations which lead to disease, or you can acquire problems during life. The inherited disorders, good examples are the anemias. Sickle cell disease is sort of the prototype. Thalassemia, which is a deficiency of hemoglobin production. Immune deficiency disorders are inherited as well. And then the acquired disorders are ones that are very often pre-malignant conditions, ones that are sort of pre-cancer, or leukemia is probably the best known. And we want to use sort of basic information to try to understand how these come about and what we might be able to do about it. So I've focused predominantly on red blood cells, not exclusively, but for the most part, because it's a rather simple cell. Its job is to make a lot of hemoglobin, which carries oxygen, and deliver it to tissues. And we thought by studying this rather simple cell, we could learn some principles and also perhaps apply these as well. And so red cells are sometimes known as a bag of hemoglobin. Really, at the end of their development, they're just chock full of hemoglobin. And hemoglobin is shown there as a tetramer. It has 4 chains, 2 alpha chains, 2 beta chains. This picture is of Max Perutz, who won the Nobel Prize in 1962 for doing the X-ray structure, the first X-ray structure of a protein. And this is the hemoglobin tetramer shown there. So it's really been a model for lots of science, and this is a quote from a prominent human geneticist where he had said that hemoglobin research plays a role in human biology or human biochemical genetics that's similar to that of Drosophila. So it really, there are fundamental principles that have come from studying hemoglobin, and I think that continues to the current day, remarkably so. So what are the diseases of hemoglobin? One of them is this disease, which is beta thalassemia, which is merely a disease where you can't produce one of the subunits, the beta subunit. This is the blood smear. It's very abnormal. It doesn't have sort of regular cells in it. The children who are untreated have a large sort of frontal portion of their skull because their bones are expanded trying to make blood cells that they're really unable to make efficiently. And they need blood transfusions to survive. The other disorder is sickle cell disease, which was first called, actually in 1949, the first molecular disease, because it was the first disease where one could recognize a protein as being characteristic of the abnormality in the disease. And then 7 years later, the actual genetic mutation was identified as a single amino acid change. One amino acid in 146 amino acids causes this protein to sickle, which is polymerize, form long chains, and destroy the red cell, essentially. And that's really the sort of classical molecular disease. Now, knowing that mutation, which was more than 65, 70 years ago, for the most part, unfortunately, hasn't helped patients, which I think gives you a sense of the magnitude of the challenge of taking science and bringing it to the patient. I think one of the other important aspects of the hemoglobin disorders is that they're not rare. They're sometimes called rare, particularly in the U.S., I'm sure in Canada as well, but there are more than 300,000 births a year of children with these conditions, and most of this is in under-resourced countries, sub-Saharan Africa, parts of India, South America, and as one gets infectious disease under control, particularly malaria in sub-Saharan Africa, children who would die in the first 5 years of life will survive, and so the actual prevalence of patients will increase over time. There are several million, 5 million. It's really, I think we don't know the exact number at this point. And why is there an area that this predominates? Well, this is a figure of the globe showing what's called the malaria belt, and I think that's a mosquito on this malaria belt, and the reason for that is that carriers for sickle cell disease and carriers for thalassemia are selected they have selective pressure under the presence of malaria because the malarial parasite doesn't grow as well in the carrier cells for sickle cell disease or the cells for thalassemia. So in the last portion, I'll just sort of cover some of the science to give you a sense of how the discoveries have gone. So we've been interested, obviously, in how these cells develop, the red blood cells, and so the paradigm is that DNA, contains the genes, and you have transcribed the RNA, messenger RNA, which you hear from Katalin, and it makes the proteins. And we've been interested particularly in how the genes transcribe, and they're read out to this RNA. And we really have wanted to know what instructs the red cell development, what's special about the genes that are expressed. And we first approached this by trying to study this disorder, beta thalassemia, where you cannot make the beta chain well. We thought that would tell us something unique. In the early days of molecular cloning, we identified all the mutations that cause this disease. And these little sticks here indicate where the mutations are in the gene. But these mutations are kind of generic. They affect any aspect of gene expression of virtually any gene, but just happen to be in this gene. And that doesn't really tell us much about the red cell. But knowing the mutations that cause this disease was very important in prenatal diagnosis. And this is a summary of other people's work from the island of Sardinia, where as many as 20 or 30% of the population are carriers for beta thalassemia. And once the mutation was known on the island, it was possible to do prenatal diagnosis for this disorder. And it's a little hard to see in the red. I don't think the pointer will show. Actually, there it is in the red. The incidence by the 90s was essentially zero. And so really the disease, new births were eliminated on the island, which had an enormous impact on really the health and the burden of this disease on the island. And So knowing the mutations can be exceedingly important. So how do red cells genes or how they're expressed? Well, we learned thereafter that there was a master regulator, a transcription factor. This is a protein that is expressed in the nucleus and binds the genes that are expressed in red cells. And this protein really makes a progenitor cell, an early cell, become a red cell. This is the structure, this is the sort of cartoon of the protein, and here it is binding to DNA. And every gene in that's important for a red cell is regulated by this gene. But I won't go through all the details, but the point was that that gene is not specific for any stage of a red cell. It's required for every stage in development. And that's important because we don't have one hemoglobin. We actually have several hemoglobins during our development, from really embryonic life to fetal life to adult life. We change this beta-like chain, and that's because on a chromosome 11, we all have an embryonic gene, 2 fetal genes, an adult gene. And the adult gene over here, the beta gene, is where the sickle mutation resides and where all the mutations of thalassemia reside. But we switch around the time of birth from depending on the fetal gene to the adult gene. And as adults, we have about 1% of, and we wanted to understand this switch. And why would you want a fetal globin or an embryonic globin? Well, the reason is in the baby, the fetal hemoglobin has a higher oxygen affinity than the adult hemoglobin, which allows the developing fetus to extract oxygen from the mother's blood. And that's really sort of the evolutionary reason why we have a different hemoglobin. This, back in 1948, this pediatrician observed that those children who would have sickle cell disease around the time of 6 months of age and thereafter were not sick at all in the newborn or neonatal period. And what she postulated at that time was that fetal hemoglobin was protective. In other words, fetal hemoglobin would protect you from having the sickle cell phenotype. And why is that? Well, the way things work on this chromosome is you express only one gene at a time. So if you express more of the fetal gene, you express less of the sickle gene. And so you have less of the sickle protein. And also, fetal hemoglobin blocks this sickling process, this formation of these rods that destroy the cell. And so for probably 4 decades now, it was postulated if you could raise this level of 1% to about 20% fetal hemoglobin, that would essentially be curative for patients with sickle cell disease or beta thalassemia. And essentially that's been the vision and the goal for the last 4 decades. And so the question has been, how do you switch from expression, there we go. How do you switch from these fetal genes, shut them off, and turn on the adult gene? Sounds like a simple problem, but it's taken many years to work out. Started thinking about that years ago and really had a difficult time. And that's when new technology came up. This is the point, you need to be flexible. And the new technology was called genome-wide association, which is study of common genetic variation. And it's rather simple. It's sort of looking now, since the Human Genome Project, we know the sequence of a sort of common human sequence. You can ask, where is there variation, where there are changes, across that whole sequence and try to correlate that with particular traits or diseases. And that was done simply by looking at the level of fetal hemoglobin in people, just like everybody in the room, and correlating that with a place in the genome. That was done in a couple of studies. And there are 3 places, 3 chromosomes that associate with the level of fetal hemoglobin. We knew about 2 of them, and I won't mention anything about these at the present time. But the new one was this one, this protein called B11A, and that's really the one that regulates largely the switch. And how do we know this protein's important? Well, we could take advantage of other technology, which is to take mice that have been genetically manipulated. And in this case, there are mice that people have made that essentially phenocopy sickle cell disease. So these are mice that have a human gene that has a sick mutation in it. These mice have sickle cell disease, have vascular problems, they die early, and we could mate these mice with other mice that had a mutation or a knockout, is called, of B11A. We could knock it out just in the red cells by genetic tricks and ask what happens. So this is the starting mouse, the sickle mouse. It has these cells that are abnormal, which are the sickle cells. And if we take B11A away, the cells look normal, and everything is fine. They just make a lot of fetal hemoglobin. So just changing this one gene is enough to eradicate the disease. And then really the question was, what is it about that? To make a long story short, we could pinpoint exactly where that genetic variation was that was critical. And it occurs within the B11A gene, within an intervening sequence or an intron. This is a non-coding portion of the gene, but it's a portion of the gene that's important for expression of this gene within red cells. So it's called an enhancer. And what was more surprising was there's an Achilles heel, or one place in this enhancer. The enhancer is 10,000 bases long. You just need to cut in one place, one small sequence within 10,000 bases, basically cripples that enhancer. And if you cripple the enhancer, you express less of the gene, B11A. B11A inhibits the production of fetal hemoglobin, and you make more fetal hemoglobin. And so this is what's been put into practice now in clinical trials using Cas9 CRISPR editing, which you've probably heard about. You just edit this site, you cripple the site, you make less B11A, the fetal genes are no longer repressed or shut down, and you make more fetal hemoglobin. So the journey has been remarkable in the sense that going from the whole universe of DNA in our body, 3 billion base pairs, we now know specifically a protein, how it works, and exactly where to block it, if you will, to turn on fetal hemoglobin. And how is that done in the patient? This is the kind of experiment, the clinical trial that's done. Take an individual here, and you remove what are called hematopoietic stem cells, these cells I showed you earlier. You take them in the laboratory, so it's not in the patient, in the laboratory. You modify these cells, you take the modified cells, and you put them back in the patient after you've destroyed the patient's own bone marrow, which is critical because then you give them back their bone marrow. Essentially, it's now modified. There are many things that are important in terms of how to do this, but it's all critical, but not important for this discussion. And here's the first patient that was treated. This is not our work. This is actually two drug companies, CRISPR Therapeutics, a new company, and Vertex Pharmaceutical. And they treated a patient just the way I described. And here at the end, after 9 months, this patient has 46% fetal hemoglobin, which is more than enough to prevent sickle cell disease, and she's perfectly well. And they've done this now many times. They've reported publicly here 31 patients who've been treated, and they're all well. And this therapy will be approved most likely in the first part of next year by the FDA in the United States. So it'll be the first CRISPR editing trial approved for therapy. So I'll leave just a few messages about the science that, through really a long series of experiments now, we understand how this switch occurs, and it's a fundamental physiologic switch that we all have, and we now can explain it in molecular terms. The clinical trials that have gone on in both sickle cell disease and beta thalassemia have been really very successful, and all the patients, including those now with thalassemia, are essentially cured. And it shows that this is an effective and tractable, we can do it, procedure with disease-altering outcomes. In other words, these patients are now well. And this is validated. I think this gene is validated. But the importance of that is we can do other things to make it easier to treat patients going forward, and that would be the future. A complementary approach is to develop small molecules or drugs that will do this, and understanding the mechanism of this process, we can at least think about that now. And really the goal is trying to expand the therapy to make it easier, less expensive to really treat many, many more patients. Just want to make the point that many, many people do this work. It's not myself, it's trainees, it's over a long period of time, and I think that's really a common thread of all our work, that no one individual does it. It's a collaborative effort taking really many years. And I'll stop at that point and be glad to take any questions if we have the time. Yeah, okay.
So we do have time for a few questions. There are people with microphones going around, so I see some hands up already. If we can get the microphone. We don't have a lot of time, so please be brief with your question, and we'll do our best to answer or address a few. There's one down here in the middle.
Hi. Dr. Perlman, my name is Yael. I'm from the Ontario Science Centre Science School. So I know that right now you've had a lot of success, and I think it's incredible how many people benefit from the treatment. But before that, I know was a lot of failures in the trials and a lot of difficulties. So I was wondering how, as you're studying something and you're working with people who will develop illnesses and diseases, how does that affect you on a more psychological level? And how has that affected your commitment to the field and what you're studying?
Okay, I'll try to answer that. We do mostly discovery, and I rely on others to do the translation, as the treatment of the patients. So I'm not directly one-on-one with the patients. However, I think we do this work because we want to make the most positive outcome we can. And I always say that if you can provide patients with something that's really transformative, then they're going to want it, they're going to accept it, it's going to be expanded in its use, and you'll get the gratification, obviously, as well. But if you provide something that is not so good, sort of a half-baked solution, they're not going to be very happy, and you're not going to be very happy, and it's not going to be taken up. So I think it takes time to develop, something that's really effective and transformative. And I think that's sort of what's happened over the years. We've had for sickle cell disease, there's a pill you can take. There are other things that have been done. They're beneficial, but they're not transformative in the sense that the individuals still suffer. And I think these new therapies will offer, just a radical change in their life, and I think it'll be accepted.
Thank you.
Yep. What's the average age of patients that are treated? Yeah, so in these kinds of clinical trials, because obviously it's a trial and you don't know it's successful till you've executed it. The review boards, those who approve clinical trials, mandate that first they be adults, and then if you see some positive benefit, you can move to younger adults, adolescents, and eventually younger children. So that's sort of that's the progression. Ideally, you'd like to treat as early as possible to prevent any complications that might be irreversible, which can happen in sickle cell disease. One can get a stroke, and it can happen in early childhood. So you'd want to treat before that occurs. And but when you start the clinical trials, you can't start in young children. So there's going to be there's a progression, if you will. And I think first adults are treated, and then younger children.
So probably got time for one more question. That's probably all.
Thank you. So I find it fascinating that the fetal hemoglobin version is protective against sickle cell anemia. And it's interesting that there are these experiments being done or there's this effort to increase fetal hemoglobin in adulthood. Are there any negative consequences to increasing fetal hemoglobin?
Sorry, I should have mentioned that. There are absolutely no consequences. There are some very rare situations where there are deletions within the complex on chromosome 11 where individuals can only make the fetal product. And 100% fetal hemoglobin is perfectly compatible with normal life, and even, surprisingly, with mothers who then will have a baby, even though the baby has the same oxygen affinity, it's fine. Now, the circumstance, if you went up to Mount Everest at a high altitude with low oxygen and you did a marathon, you might have some problem. But besides that, it's perfectly compatible with normal existence. So the more the better in this case.
Okay, well, I'd like to thank Dr. Orkin, and I think we do have to move on. So Brittany will introduce our next speaker. So thank you very much, Dr. Orkin.
Okay, so it's now my pleasure to introduce our second speaker, a pioneer and advocate for the field of RNA biology, Dr. Katalin Karikó. She earned her PhD in biochemistry at the University of Szeged, and she worked as a postdoctoral fellow at Temple University in Philadelphia prior to moving to the University of Pennsylvania, where she now serves as an adjunct professor. Dr. Karikó is the Senior Vice President at BioNTech RNA Pharmaceuticals and has earned countless national and international distinctions in the past few years alone. She is the recipient of the Princess of Austria's Award, Albany Medical Center Prize in Medicine and Biomedical Research, Breakthrough Prize in Life Science, and this year is a recipient of a Canada Gairdner International Award. Dr. Karikó, alongside Dr. Wiseman at the University of Pennsylvania and Dr. Cullis at the University of British Columbia, have been distinguished this year for their innovative work in mRNA modifications and lipid nanoparticle delivery of mRNA into humans. This groundbreaking research is what allowed for the rapid generation of the highly effective mRNA COVID-19 vaccines. The mRNA modification and lipid delivery system prevents cellular inflammatory responses and subsequent RNA degradation. This technology has seemingly endless potential for other vaccines, therapeutics, and gene therapies targeting various infectious diseases. Dr. Karikó is an inspiring researcher, particularly to women in science, for her resilience in the face of setbacks, which I expect she may elaborate more on in her talk, Developing mRNA for Therapy. Please join me in welcoming Dr. Katalin Karikó.
Thank you very much. Good morning. I have a daughter who I remember not a long time ago, she was in high school. And I know that the attention span is very short for the high school students. And I remember also, that I tried to encourage her to to get excited about science. And sometimes I was scientific journal, I showed her an article, and I said, isn't that interesting? And, she read, and she said, that's okay, Mom. So I understand that. So, but I hope that some of you will put down the phone, turn over, because I know that very exciting and important text message is coming, and you have to answer it, but if you get the vaccine for the Pfizer, BioNTech, or the Moderna vaccine, you might hear something about how it was developed. So I am talking about how the mRNA was developed, and also about my journey, how I got here. Yeah? And the messenger RNA, you heard this word, yeah? If you get the vaccine. Did you get mRNA vaccine? Yeah? Okay, so that it was mRNA was discovered more than 60 years ago. And it took 60 years to develop the mRNA to FDA-approved product in the form of LNP mRNA COVID-19 vaccine. So what happened during these 60 years? And there were some important discoveries, but most of the time as science, just incremental increase, improvement, and this is what happened. That's how science works. So by 61, we learned the basic thing, how information goes, and that's the information flow in every cell in your body and every free living organism, that from the DNA, there is this RNA is copied, and it comes up to the cytoplasm, and here the ribosome is the protein synthesis factory. I heard that, you are just right now is in a biology class also, yeah? So we have to talk something about biology, and you know about ribosome, and the ribosome, the protein is made and maybe secreted or maybe intracellular. But scientists in the 50s spent a lot of time to find what is this molecule, because it was immediately degraded. And one important thing was that, scientists couldn't make, in 1961, they couldn't make an mRNA. So they could just isolate and study and so on, but I don't go into this. I mentioned just one important experiment when scientists, 1984, discovered how they can make in a tube, how they can make mRNA. Without their discovery, you wouldn't get the mRNA vaccine, because this was made in a tube or maybe a big bag, actually 60-liter bag. Yeah? So they have the plasmid DNA. There's the double-stranded DNA, and they use the phage RNA polymerase. The phage is the virus of the bacteria. Can you imagine bacteria had to worry about the viral infection too? So the phage is has an RNA polymerase which has high fidelity and can copy where the DNA. And then these scientists at Harvard University, actually, they generated an RNA which coded for interferon. And they injected it to frog oocyte because we are in 1984, there were no cell culture and things, and the frog oocyte is a large cells, and they can inject with a capillary. And what happened when they cultured these kind of injected cells and took the medium, and they found that the interferon, which has an antiviral effect, was really antiviral. So this was first time that somebody made an RNA by design, and that RNA was translated in a cell and produced the desired protein. So this was 1984. So a lot of things happen, and with how I got research involved, this was one experiment I am telling you. This was actually created the basis for the Moderna, BioNTech, and Pfizer vaccine. And what happened is that with my colleague, I made the messenger RNA, and he put this messenger RNA to a cell, which is called a dendritic cell. This is the immune cells. This is the professional antigen presenter cell, the teacher cells of our immune system. And what happened is that when the mRNA I made in the tube, just like those colleagues in 1984, what happened was the RNA was very inflammatory. This purple shows here, and the measurement of inflammation was measuring TNF-alpha secreted by these immune cells, so it was very inflammatory. But when we isolated out different kind of RNA from the cell compartments, what we found is those were less immunogenic. What was important is that we discovered that one of the type of RNA, the tRNA, transfer RNA, you might learn at school, that this carries the amino acid during the synthesis, was not inflammatory. And we knew that transfer RNA has a lot of nucleoside modification. And that gave us the idea that maybe modifying the nucleosides can suppress the immunogenicity of RNA. And to test out, we have to prove it. And how you prove it, you have to make somehow modified nucleoside-containing RNA. In science, always something come up how we should do it. Nobody has done before. And so eventually, in your body, all of the RNA is made from the 4 basic nucleotides. And then when it is making or post-transcriptionally modification introduced by different enzymes. And you have hundreds of different modifications, which is in your RNA. But we couldn't have these enzymes. Those were not even known. So what we have done, we purchased already modified nucleoside containing triphosphate building blocks. And when we generated the RNA, what we discovered is that when it contains uridine, which was modified anyway, then the RNA did not induce the inflammatory molecule. So this was the basis of, to make RNA which would not contain uridine, because it has uridine. You can see in the blue that it induced a lot of inflammatory molecule. But when we made these changes in it, then we have RNA which is non-inflammatory. And what happened is that, of course, I wanted to develop mRNA for therapy, so that the mRNA would code for a therapeutic protein, and my colleague Drew Weissman wanted to make vaccine. And of course, in both cases, you need the RNA to code for a protein, and it has to be translatable. And when we tested out a pseudouridine-containing RNA, translated and generated, we could capture 10 times more protein. So it was something for me, it was the icing on the cake, because now finally I can have an RNA which is non-inflammatory and translates so well. Because that was for years the problem why other people could not proceed with RNA therapy. Because the protein was very small amount, very short period of time was made, quickly degraded, and it was inflammatory. But now it seems by 2008 we solved all of these problems. And then comes, the Canadian scientists in Vancouver. There's the capital of lipid nanoparticle experts. And so they come up with a formulation which was very critical for the vaccine development as well. And these contains different kind of lipids. That's what, if you get the BioNTech Pfizer vaccine or the Moderna vaccine, is that these 4 types of lipid is mixed with the RNA, and that's how it was delivered. And one of the lipid, actually, the immune stimulant, because when I was in school, we learned that how our immune system works is recognizing what is foreign. That's not true. What our immune system is recognizing, whatever is causing danger. So injuries, because we have a lot of bacteria and we are very happy to have them in our intestine because they are part and making important things for us. So anyway, that all of this formulation and other things resulted that 60 years, what I told you in 10 minutes or 6 minutes, resulted that in 2020, you remember very well, in January we learned the sequence information for this coronavirus, and by the end of that year we get a vaccine. And I have to say that if this pandemic would come 20 years before, that would not be enough to get sequence information, what was in January, what we learned, because genome synthesis was not solved yet. So you couldn't order genes. It was 20 years ago when German scientists started to establish companies and you could order genes, for example. So that many, many things, of course, you couldn't read the sequence information on the internet because there were, was not available things, so that technology, different level, culminated that we have, and eventually we get approved this vaccine. So this was the science in a nutshell. So I don't bore you more with that. So I'm just telling you about myself, how I got here. So I came from Hungary, and I grew up in a very simple house. It's a reed roof and adobe house, and we had just one room, no running water, no television set, no refrigerator, and no running water. So we had to go to the street to get the water, and that was the chat room, when we went there and we learned from the neighbors what's going on. So that's how we lived. And then my father was a butcher, and I had an older sister who was good in school, so when I went to school I had to perform better because she was already good. And so in elementary school, that I was competing with different kind of chemistry, biology, and other competition. And actually, when I was in grade 8, I was 3rd best in the country in biology, in Hungary. Yeah? And, but this is also interesting because, for the competition was a whole week in Budapest, which was 150 kilometers where we lived. And, my parents couldn't afford to come with me, so they put me on the train to go to Budapest by myself. And then they learned that I had 3rd place and I did well and come back after I returned because there were no telephone. So this was, seems like, 100 years ago, but it was not, I'm not that old. So what happened is, we went to different kind of camps as a biology student. Here, everybody's, 13 years old girls. And we were competing in different territory in Hungary, and, these were the local winners of biology competition. And what is interesting is, I am talking to high school students. I should talk also to the elementary school students. These students all knew that they want to be a scientist. And here is one, that's me here, the tallest always. And then she is also a biology student. She became physician, and there are 2 of them I know that became chemists. So, at at the girls, at age 13, they knew that what they want. They wanted something. So after finishing elementary school, I went high school, local high school. The small town I grew up, it was just 10,000 people lived there, but we had a very nice high school. And here, good teachers, biology teacher, chemistry teacher, and we did a lot of experiments, actually, and ourselves, and we also sent letters. We couldn't speak English, I started to learn when I was 18, so in high school you couldn't learn. We learned Russian and French. And so what happened is we sent letter in Hungarian to these Hungarian scientists. One of them is Hans Schreyer, who's born in Hungary, but he is a Canadian scientist, and they responded, and we started to read their books, and I will tell you that I wouldn't be here if I wouldn't read that book from Hans Schreyer. But what happened after high school, because it's very, I have never been in a university, I never seen a professor, but I decided at 16 that I will be a scientist. Every day I will go to work and discover something. But at the university, they organized for the underprivileged children, which I was kind of, because my father had 6 elementary and my mother had 8 elementary education, so no formal education, and so I was not the child of professors or something. And so here we are in the summer, and we spent 2 weeks in the middle of the summer, from early morning, late night. We studied physics, mathematics, and we have seen professors, and we could see that they are just not different like our parents. So this was, for us, it was very important. Here I am. This is me. So paying attention. There was no cell phone, of course, that time. And so as a result of it, I was accepted to this university. University of Szeged is the southern part of Hungary. I went there because there was this biological research center, and that was my dream to work at that biological center. So when I was a student, I already went and then told the professor in a lipid group that I would like to work in the laboratory, because that's what I thought should start. But he said that, oh, you just go to the Fishery Institute and spend the summer there, where we fed different things to the fish, and we looked at the fat content. Like, if you feed the fish with corn, it seems like they are pig. Yeah? Because fat was very saturated. So anyway, that was, my life as a student, and I started to work in a chemically synthesized RNA. And what happens in these days, that I also married, and then, I get my PhD, and I became a mom. Yeah? And, my husband was very supportive, but still the 9 months is, on me. And so it made a little bit difficult to be scientist. But I had help from the government, because in Hungary we had affordable, high-quality childcare. So I put my little daughter to, when she was 3 months old, to this center, and there were registered nurses were there, and every day a pediatrician attended. And I could leave a message that if I could see some problem and I want to see that whether I should go a specialist or that's okay. And it was, they also get every vaccination. We had to just sign as a parent, and we agreed that gave them the vaccination. So my daughter was growing here. We were in Hungary in '94 when I lost my position in Hungary. And again, I can tell you several times I lost my position. I wouldn't be here if I wouldn't do that. So that's, you don't have to think that something bad happens. It's, maybe it will be, eventually it will be good. So we arrived to the United States here with the first car, with the Pinto we purchased, and started to work at Temple University. So those girls who are here and want to be scientists just mentioned that you can be also a mother. And then my daughter came out, turned out is okay. She became a 2 times Olympic champion for the United States. And the women 8, I might mention that we were there in the Olympic Games, and in London, the they get the gold medal, and the silver went to the Canadian women 8. Yeah, so they did very well. And of course, my daughter has now daughters, so I am a grandparent. And so it's just a family life of a scientist is just like, your own family, you have children and grandchildren. And what I was doing in these days, I was trying to encourage the young one, the younger than you are, that, to be scientists. And here is some book. This is in Hungarian, this is in English. Here is Drew Weissman, and this is me. And so, I have this life of scientist, and I thought that I can tell you a couple of things that you might consider, not even if you want to be scientist, you can be anything else. It will be some advice. First of all, I am telling you that you have to like what you are doing. So if you select any kind of job, you have to enjoy it, because when you like it, you will be good at, and you even like more. And it will be, you don't need a hobby, you want to spend all your time with that. And so I am telling you, if you like to be in the spotlight like I am right now, probably you should be actor or actress, because the scientist not many times is there. And then, if you want to be rich, then I don't know what you have to do. But if you want to be, let's say, curious about, like this morning you were coming here, did you notice that the leaves are yellow? And you wonder about, the other is red, one is yellow, and it was green. How the yellow came from the green, or it was already the yellow was there, but the green covered up. So there are scientific questions around you, always. And then you see that they're falling down. How did they know that it is fall? Did they have a calendar that they have to come down? Or you look up and you see the Canadian geese coming, going. How did they know which direction? So that you are curious and you want to know. Yeah? So many, many things around you. And so that's, if you like to understand the problems and solve it, and eventually, as you heard from Professor Orkin, is leading to that you cure people. I mean, that would be the most fulfilling life you can have. And so it is very important, to love your work. And I suggest to you that, be modest, that just be yourself. I like to be in this left, that kind of carrot. I want to be not like that one who's showy and so on. I mentioned you that Hans Scheyer, and I don't know whether you heard, but Hans Scheyer, he was coined the word in the 1930s, stress. Prior to that, this word was only in the physics. And he demonstrated that in animal studies that the stress can kill. So and then human can be killed by the stress, but not necessarily just how you perceive. And he also said that you need stress, positive one. So this morning you woke up and you get out of bed because you said, oh, today we can hear Professor Orkin and Kati is coming, and the excitement, the anticipation, all of these things which makes you, move and come and be here. And so you need positive. But how you make the negative stress to positive one is what at age 16 I read this book about the stress, and then I told you I wouldn't be here if I would not read that. And it was very important. Listen, you have stress. Do you have stress? Yeah, okay. So you have to learn how to handle. And what important thing is that whatever you think through a problem which causing your, this pressure and something, has to be ended with what I should do. Okay? You can say that, oh, the teacher should give better grade. I said, you cannot change that. You can think, oh, my parents should do the tatam. Yeah, you in that age you think a lot of times that, but you cannot. You cannot change others. It's stressing yourself to wish for that. So instead of you have to say, okay, I want better grade, I should study more. I should be more prepared. Every time you have to find what you should do. and not other person should do. And it is also, I have to say, that so many of us, blaming others, because it is so easy, oh, because of the, I don't know, the mother, my neighbor, my friend do that, and, and blaming them. But, if you blame others, again, it won't help you, because then you say, okay, it's not them. And that's what stresses you more. And it is, so I mentioned that I was terminated in my position several times. And what is important is, instead of feeling sorry for myself, feeling that, licking my wound, that, I was better than the other, and why me? When somebody is making a decision and you cannot change that, then you have to spend all of your energy, what next, what I can do. And in a setting like, in a workplace, or even here, you can see that, oh, the other student, maybe you feel that they don't study that well, and they still get better and tararam, and it makes you upset. Again, you cannot change that. You have to focus on what you can change, and you can be better. And at the workplace also, you can see others are promoted, higher salary. If I would have paid attention, I wouldn't be here, because that's what happened always. Others always get probably higher salary, but if I pay attention to this one, already it was a distraction, because whatever is in front of me, and when I demoted from my position at University of Pennsylvania, I really loudly said, I am in America, in an Ivy League school. The bench is here. Who cares that I am not professor anymore? Where else, if not here? So you have to always focus on what you can do, not wishing that somebody else should do. So then you have less stress, and you will be happy. Not saying that, you are kicked out of job and you say, whoo, that I'm kicked out, but you have to just, focus on what next. So the next one is, to believing in yourself. So you will go to, right now maybe you are the best in the class, and but you are going to the university or going in a workplace, anywhere you go, you realize that everybody is better than you, and then you feel depressed that you were the best and no, you are not. Because, that you, and then many times, you just lower your bar, and then you say, oh, okay, I might be just an assistant to somebody, whereas previously you wanted to, proceed and have a more qualified job. And I have to say that, coming from a small town, like I was, and going to the University of Pennsylvania, as an Ivy League school, 30,000 employees, everybody's, perfect English, not like mine, and everybody knows so much. To me, imagine that I would think about something they don't. I had to believe that, because otherwise I would say just I can assist them, okay? So it is, here is one saying that believing in yourself is an endless destination, but if you believe that you will fail, don't even start, because that's your end of your journey. And of course, life, it always seems like, a very smooth sailing or biking, and then just, on the way you realize that, oh, there are so many problems, but, as long as you have set up your goals, then, you work towards it. It is also very important, that you are in a comfort zone. Right now, mostly not much worry. Your parents is taking care of you, have your home, you don't worry about mortgage to pay, and other things. And then you are in a comfortable place. And you have to expand your comfortable zone by exploring other things, and you have to get out from it, And for me, with my 2.5-year-old daughter and my husband, getting a one-way ticket from Hungary to Philadelphia, not even knowing the person who I will work. I didn't even met a person who met that person, so that I had to ask how I recognize him, and he said he's 6 feet tall. I had to calculate, because I didn't know at that time, in Hungary in centimeters, so I didn't know that he will be a short man or a tall one. And so it is, coming to a place, and then I had no relative, our currency was not convertible, so that I can see that most of you are not in a native Indian here, so your parents or yourself, came from another country, and then it changed you in a way, because mostly your parents had to, go through as an immigrant struggle with things, and they were, persevered because they had a dream, and it changes you. They asked me that if I would stay in Hungary, wouldn't be able to do all of these things which I discovered. Maybe, but there were not as pressure as when we came here. And we arrived, and in the evening and next day I went to work, and so it was with a hard time. But we had to left our comfort zone and come and explore. And here, finally, coming back to my daughter again, when she was here in the picture, she was 7 years old. She was in 2nd grade. And she came home from school. The school year was over, and she started to write a letter, and I asked, who are you writing? And she said, oh, Mrs. Wilson, I want to thank her for this wonderful year, how much we learned. And she didn't see this one from me, because, I have never wrote one. And then I realized, that I should, thanks to all of those people, the teachers and the people who helped me on my journey. And so one thing is that, you can learn from everybody, anybody, even a 7 years old can tell you something. And the other thing I would like to ask you that from time to time, please say thanks to those people, the teachers, your friends, or your parents, what they have done for you. Thank you very much.
Well, thank you very much for this exciting journey. And we do have time just for a few questions. I think the schedule is a little tight, as I say, but if there are some questions, get the mics around, and we will try to get as many as we can if you're pretty concise with your questions.
Hi, I have a question about pseudouridine. Does the presence of pseudouridine in mRNA affect the protein fidelity, for example, like 3D protein structure, or does the protein remain exactly the same, completely unaffected?
So when we discovered that putting pseudouridine makes it non-inflammatory, it turned out 10 years later that our mRNA in our body also has pseudouridine. And a recent study demonstrated that the 1-methyl-pseudouridine, which is in the vaccine, actually increased the fidelity of translation and also the fidelity of transcription. So when you generate the RNA from the plasmid, the fidelity when they identified that compared to the U-containing RNA, it was higher, and the translational fidelity was also. So it doesn't change. The pseudouridine has the same hydrogen bonding that the uridine, and it is also present in our mRNA, not changing. The short is not. Short answer.
Okay, there's a mic coming.
Hello, I'm just wondering, how do you get the messenger RNA through the phospholipid bilayer? Because I assume it's a big molecule, so I'm just wondering how you get it through the bilayer.
So the messenger RNA is how it produces. First, it in low pH, bring together in an ionizable lipid. When this lipid is positively charged and the RNA is negative, and then you put the 3 additional lipid to it, and then you have this particle. And when you put on the membrane, the cells had endosomal activity, so they pick it up. Mostly, actually, for the vaccine, they also showed that it was macrophages and dendritic cells. So immune cells will pick it up, and not the muscle cell, actually. And they pick it up, and when it is an endosome, the pH in the endosome is going down, and then it will fall apart, and some is escaping to the cytoplasm.
Thank you.
Here she is, she was so tentatively listening.
In the front row.
No texting. I would want. Here, show.
So I was wondering if you've ever felt like you've had so much work to do, just completely overwhelmed, and how you dealt with that. And also, for all the people that didn't take the vaccine because they didn't understand or believe in the science, what do you wish you could say to those people?
So getting first answer for I recognize as a scientist, I like to talk to other scientists, not to reporters and try to explain, and that was wrong. We should have, but now that I told everything should be what I should do, no, so I am talking last night also the student reporter asked me, so every time I will respond, because the reporters and you and me educate the public about, because many people will not take the vaccine because they didn't understand, they didn't know. And genuinely they wanted, there are some people, okay, who would never ever can convince, but other people just wanted to understand and learn. And we have not done with the reporters. And listen, if I ask you, can you name a Canadian scientist who's still alive today? How many of you can name somebody? Okay. I cannot see anybody. Okay, so that's the problem. You can name the hockey players, and why they are more important than the scientists, yeah? And so who is responsible for it? We as scientists and the press. But the press says, oh, people want to read about them. But if they would write about us, maybe people want to read about us, yeah? So, and the first part was, the question was, what was? Yeah, I mean, we always felt a lot of things to do, but that you break down what your plan is, and then you prioritize, as Professor Orkin said that, and then you do these things like that. Of course, that sometimes I have to prioritize. I am here with you, and my daughter is due in any time, getting the second grandchild. But, then after that, the family will be prioritized, and I will be with her. But right now, you are the priority number one.
Okay. Well, I think we probably should wrap up. All of us have different commitments. So I'd like to thank the speakers again, thank Brittany for introducing. Thank you for coming, and I hope this was a stimulating event for you, and that your teachers go back and your classmates next year come. We hope we can keep doing programs and not be interrupted by having to do them virtually for 2 years and be back in person in a venue like this. So thanks everybody for coming, and I think Kate has a few words to get people going in the right places and the right direction for the rest of your stay at York, and I hope you enjoyed the morning and enjoy the rest of the day. Thanks.